the D-Mice Research Conference, which we tend to have a lot of at the end of the term. Today's speaker is Chris Klocke, who is a PhD candidate and National Library Medicine Fellow in our program in the Bioinformatics and Computational Biomedicine major. He has a Bachelor of Science in Biology from American University and worked in biotechnology development before starting the program. He currently has research interests in the genetics of cancer and tumor evolution. So he is presenting his symposium, which is a review of the science and the literature of his research area that will eventually become his dissertation proposal. He'll be back to defend the proposal down the road and then ultimately defend his dissertation and then graduate with a PhD. So I will turn it over to Chris.
Thanks, Bill, for that great introduction. Can everyone see my slides all right?
Yep.
Okay. With that, I'm going to get started. And as you mentioned, I'm going to talk a little bit today about intra-tumor heterogeneity and drug response. And more specifically, how we can potentially uncover subclonal drug resistance using various single-cell technologies. First, I just take a minute to talk about really why this is a relevant problem to study. So, if we were in a situation where, routinely someone was diagnosed with cancer, they were given a treatment, and they reliably showed durable response, and that was the end of it, then this idea of tumor heterogeneity might not more than a question of just trivial biological interest. But the current state of things is that often when someone's treated for cancer, they might have a tumor that's treated with a drug. Often they show drug resistance and treatment failure, and this leads to a huge amount of cancer-associated morbidity and mortality. So to address these limitations of the current state of the field, it's important to study one important factor in drug resistance and treatment failure, which is tumor heterogeneity. When cancer researchers discuss heterogeneity, they usually talk about two different types. So there's inter-tumor heterogeneity, which is differences between patients, and then there's intra-tumor heterogeneity, which are differences that occur within a single patient's tumor. And for the sake of this talk, I'm really going to focus in on the second type of tumor heterogeneity, intra-tumor heterogeneity, which is that difference within a single patient. Earlier on in the 20th century, there was a more classical view where you can think of a tumor that's composed of a population of cells. There's an earlier view that these cells might be more homogeneous, but this has really given way to a more modern understanding that there are these distinct subpopulations of cells that make up a heterogeneous tumor. In addition to this, there's this idea of clonality and the clonal nature of cancer. So when we think about cancer, we think of these populations of cells as deriving from a common ancestor. We can actually see this heterogeneity visually. So in this image, which is an immunofluorescence microscopy image taken from a 2016 analysis that looking at a breast tumor sample, you can actually see through these fluorescent signals the differences between cells within the tumor. So the top arrow we can see is pointing to cells, and there's some of these light blue cells that express. So they're really looking at three different receptors here. These are three proteins that are important in classifying the subtypes of breast cancer. So this is the estrogen receptor, the progesterone receptor, and the HER2 receptor. So we can see on top the top arrow pointing to a kind of a sub-region showing a lot of these light blue cells that express both the progesterone receptor and the estrogen receptor. The middle arrow in this population, there are more cells that express just progesterone, just estrogen but not progesterone, and on the bottom, the reverse, just the progesterone but not the estrogen receptors. So while we, often subtype patients of, oh, this patient has a an estrogen receptor positive breast cancer or something like that, this actually can vary within that patient's tumor. So this is, just again, a way we can see heterogeneity visually. But before we had these, more advanced modern ways of looking at heterogeneity, we kind of have to take a step back in time and look at where this idea started. So in a 1976 science paper, Peter Knoll put forth some of his own ideas, and then he also kind of summarized some of the current thinking in the field at that point. And he put forth this hypothesis of sequential selection of mutant subpopulations of cells as a model for tumor evolution. This was an important step forward. And he also, just kind of going back to this idea of clonality and the clonal nature of cancer that I mentioned above, he also suggested that the cells in a tumor appeared to be derived from a common progenitor. And the experimental data that led him to draw this conclusion was that he saw these abnormal karyotypes. So there were these cancer cells that had these chromosomal modifications, and he saw the same abnormal chromosomal pattern in many cells within a tumor, suggesting that they did actually derive from a single ancestral cell. Now, one just last note to mention here is that Knoll was actually a little bit ahead of his time in this paper in suggesting that these subpopulations of cells, these mutant subpopulations that are being selected for, could actually be driven by genetic changes, which is the more kind of canonical way to think about it, but then also by other changes such as epigenetic changes, which is something we think about now. So this was pretty interesting that he was thinking about this back then. Now, just to think about this visually, you can picture a normal, potentially precancerous cell that sustains some type of an alteration. This could be a mutation that seeds a founder clone of a tumor cell population. Now, this parental clone may contain cells that then sustain additional changes, leading to the development of different distinct subclonal populations of cells. As we can see here, there are actually multiple different mechanisms that can create these distinct subpopulations of cells. So on the top, we see genetic subclones. This might be a subpopulation of cells that sustain some additional mutation or genomic rearrangement. There can also be things like DNA methylation. This gets into the epigenetics. So you can imagine a gene that has DNA methylation in a promoter region that leads to different expression patterns. So this population of cells could be distinct without actually having any changes in the primary nucleotide sequence in its genome. And then additionally, you can have things like, so in cancer biologists think about this concept of intrinsic versus extrinsic factors. So in the intrinsic factors, you can think about genetic and epigenetic changes that are going on within a cell. But then there are also these extrinsic factors, such as, nearby cells in the immune microenvironment and different parts of the microenvironment that can be secreting various factors that change the behavior of a subpopulation of cells within the tumor. So just to recap this, there can be these distinct subpopulations of cells that make up a heterogeneous tumor, and they can come from multiple different mechanistic sources. So this could be genetic, epigenetic alterations, these intrinsic factors that I mentioned. But then it's also worth mentioning other reasons why heterogeneity might come up in the data. So in addition to these kind of distinct subclonal populations we're interested in, in order to study these concepts, you have to perform experiments and look at data. And we might actually see heterogeneity in the data coming from other cell types like tumor-associated fibroblasts, various tumor-infiltrating immune cells. There can also be tumor purity issues where you have a sample that is contaminated with healthy cells that are adding noise to the signal. And then you also have this concept of cancer stem cells, which I'll just briefly discuss a little bit more. So the idea of cancer stem cells is really a hypothesis. This is an open question in the field. It's not one that I attempt to tackle head on. I'm just more mentioning it as a potential source of heterogeneity. If you ask 5 different cancer biologists what they think about cancer stem cell hypothesis, you'll probably get 5 different answers. But this is really just the idea that just as you have stem cells in healthy tissue that are capable of differentiating into and seeding, the bulk of the tissue, and they remain relatively undifferentiated, some proposed that some tumors contain these cancer stem cells that remain in this less differentiated state. They have a little bit different of a character, and they are capable of seeding the rest of the tumor population. And this has actually been observed experimentally in some tumors. But really the idea being, going back to the idea of heterogeneity in the data, if you have different subpopulations of cells within a tumor that are on different places along this differentiation hierarchy, this can show up as heterogeneity within the data as well. So just to recap really what I mean by subclones, this must be experimentally defined. And if you're thinking more about hematologic malignancies, this could be a subpopulation of cancerous blood cells within the bone marrow. So we would think about this more as being likely to be spatially intermixed. Whereas with solid tumors, this could be a subpopulation of cells within the tumor that actually could be spatially distinct. So, this poses a challenge. You can imagine if you take a sample or core biopsy or something of a solid tumor, and you look at the subpopulations of cells present in that sample, you could be looking at that, but then there could actually be some subpopulation of cells elsewhere in the tumor that you're missing. So this spatially distinct subclones, is an important concept. But then, they could also there could be cases where they're more intermixed. My primary interest here is really subpopulations of cells as they manifest at the level of gene expression. So just to kind of tie this back to the motivation slide that I mentioned at the beginning, we do have this problem of drug resistance and treatment failure. So the reason why it's of clinical importance to study these heterogeneous subpopulations of cells is that potentially there are these, there's the bulk of a patient's cancer that might be responsive, but if you have a subpopulation that is distinct in some way where it's capable of driving drug resistance and relapse, then there's really a clinical importance to understanding that population of cells. To think about this visually, you can look at the light blue cells on the left making up the bulk of a tumor and think about these as being drug sensitive, whereas there might be in the dark blue a subpopulation of these cells that is resistant. So this is really akin to Darwinian evolution, where you have a population of organisms and then you have various environmental selective pressures that cause evolution. So really, you have the treatment. You can think of the drug as a selective pressure that is going to affect the evolution of this tumor population in various ways. So you can think about the drug as being the selective pressure, and then over on the right, when this subpopulation is selected for and expands, you can think of this as being drug resistance driving treatment failure. Now, to make this a little bit more concrete, I'm just going to walk through an example of a cancer type and a drug that has a long history of study at OHSU, which is chronic myeloid leukemia and the drug imatinib. So chronic myeloid leukemia is a blood cancer where in the majority of cases, you can see over on the left, chromosome 9 and chromosome 22. Chromosome 9 has this ABL or ABL1 tyrosine kinase gene. And then chromosome 22 has this breakpoint cluster region gene. So if you get this specific genomic rearrangement or translocation where part of chromosome 9 fuses to part of chromosome 22, and parts of these two, the BCR and the ABL gene, actually fuse together, you get this, what's called a Philadelphia chromosome. So whereas the normal ABL tyrosine kinase is under very careful and strict control from different regulatory mechanisms, when it becomes fused to the breakpoint cluster region gene, it becomes constitutively active. And what I mean by this is that it signals in a way that becomes dysregulated and can actually drive the progression of the leukemia. So there was some work a few decades ago, that Brian Drucker had a big part in, that it had, they really were looking for a way to target this BCR-ABL structure in a targeted manner. And they came up with this small molecule drug imatinib, or Gleevec, that could act as a tyrosine kinase inhibitor by interacting with the BCR-ABL structure and and reducing its tyrosine kinase activity. So going back to 2002, when this work was really getting off the ground, they observed in this analysis, 90% of early stage CML patients would respond to imatinib, but of those with later stage disease, only 60% would respond, and nearly all of them would go on to relapse. So in order to try to understand what was driving this relapse, they actually did an analysis and found experimental evidence for intratumor heterogeneity that related to drug resistance. So they sequenced 10 different PCR amplified samples from each patient. Any mutation that was found in at least 2 out of the 10 samples was called as a subclonal modification. And of the patients who relapsed from after imatinib treatment, the vast majority of them, 29 out of 32, were found to have some sort of a mutation in the kinase domain of this BCR-ABL structure, which was somehow affecting the ability of imatinib to change the behavior of these cells. Additionally, there was polyclonal resistance detected in some patients, meaning that there were actually multiple genetically distinct subpopulations of cells that were each capable of driving resistance to imatinib with different mechanisms. Just about this visually, on the left, you can, using the same image as above, you can see the light blue cells, think about them as being responsive to imatinib. Dark blue cells harbor this kinase domain mutation. So then when you apply the drug into imatinib, which acts as the selective pressure, this selects for that mutant subclone with this alteration and drives clonal expansion and treatment failure of that first-line therapy. Just to think about this again, in the case of polyclonal resistance, as I mentioned, you can have multiple distinct subpopulations of cells that each have different, in this example, kinase domain mutations that are capable of driving resistance, and then these each would go on to persist following first-line therapy, and they would be present in this recurrent disease. Moving on to a little bit of later work, in 2008, they actually found that many imatinib-resistant subclones are still sensitive to these other drugs, these other tyrosine kinase inhibitors that were developed, dasatinib and nilotinib. And here they actually found that there were detectable mutations present after imatinib failure that could indicate the preference of one of those drugs over the other clinically. So this is really a success story. This shows really the translational importance of understanding the mutant subclonal populations of cells that drive drug resistance, because in this case, one of these mutants, tyrosine kinase modifications, this F317L, was more sensitive to nilotinib than dasatinib, whereas a different mutant modification, F359IV, was more sensitive to dasatinib than nilotinib. So you can actually start to see what the best clinical decision would be for a particular patient based on what those mutant subclonal populations of cells are that are present. I really just use the CML and imatinib example to make these concepts concrete, but these concepts really apply more broadly across a range of cancer types. For example, there are analyses that show drug-resistant subclonal populations of cells that drive treatment failure in other cancer types, such as triple-negative breast cancer. So triple-negative breast cancer is a subtype of the disease that is particularly difficult to treat. Going back to that initial image I showed you with the immunofluorescence microscopy, those 3 proteins of interest, it doesn't express any of them, which leads to the inability to use certain hormone-based therapies that could be used in other breast cancer subtypes. So this has been shown, and then so this really gets at the idea that understanding the relationship between these different mutant subclonal populations of cells within a tumor that make up a heterogeneous tumor and drug resistance and treatment failure, really understanding this relationship is going to be critical for the effective development of cancer therapies that lead to an effective and durable response. At this point, I'm going to switch gears a little bit and kind of transition to the second half of my talk. So really at this point, I've been talking about the biological background of this concept of intratumor heterogeneity and these subclonal populations of cells, evidence for the fact that these subclonal populations of cells might drive drug resistance. But in order to make progress here, we actually need to study these experimentally. So I'm now going to move into discussing some of the assays, so the ways that we collect data, and then the computational methods, so the way we analyze these data once they've been collected. The technologies that can be used to study kind of the molecular interworkings of cells have really taken off over the past 20 years or so. And earlier on, one of the key platforms was microarrays. So this is, really a set of probes that is capable of measuring the genotype or the expression of particular sites of interest. This gave way to bulk sequencing methods once kind of more high throughput, like whole genome type sequencing methods came online. And it's also worth mentioning that that there are certain imaging techniques that can be used. So fluorescence in situ hybridization, or FISH, has been around for a while, where you actually can get a spatially resolved image with expression of different genes or proteins of interest. And then just going back to that third slide I showed you with the breast cancer sample, you can use immunofluorescence microscopy to look for some of this heterogeneity in some cases. So when these technologies, particularly whole genome sequencing, were the state of the art in this field, the associated computational methods used these data types in order to study this question of subclonal populations of cells. So the methods of interest here, really developed in the past 5 to 10 years or so, tend to use variant allele frequency. So if you sequence a bulk sample of a tissue and you have these different reads that align to a reference genome, you can look at the proportion of those reads that have a certain allele of interest. And so this is, what these methods typically would do is they would, do some sort of whole genome sequencing. They would identify sites of interest, and then they would do like very deep sequencing on these particular mutations, so they could get a better sense of the fraction of those reads that had an allele of interest. And then you can't just assume that that's the fraction of cells. There are certain things you have to take into account with variant allele frequency, like copy number changes. So certain regions of the genome could be amplified or deleted. Something could be heterozygous or homozygous. The sample could be contaminated with healthy cells. So you have to take these various things into account. But then you can actually, at a certain level, get a preliminary sense of, what subclonal populations of cells might be present within a sample. So these methods actually have to use copy number data, so this idea of amplifications and deletions, and these methods tend to take the variant allele frequency as input, but then they also require information about copy number as input. And then they tend to then cluster these somatic alterations, look at alterations that are at various frequencies, and then kind of reconstruct a phylogenetic tree to determine kind of what subclonal populations of cells there. However, this approach does have some limitations. One of them being that these methods tend to only look at genetically distinct subclones. So as I mentioned before, there are those other ways that a population of cells can be distinct, such as epigenetics like DNA methylation, or different environmental factors, microenvironment factors. So these methods kind of don't really attempt to capture that. Really, just another set of limitations here is that you can picture, if you have a bulk sample, you have this sample, but it might contain cancer-associated fibroblasts and immune cells and some healthy cells and different subclonal populations from the same tumor, and you have these all kind of dumped together, and you're getting just sort of an average signal of what's going on there. So at a certain point, it's when that is the state of the art, that's what you use, but this really lacks some resolution to really fully understand these distinct subpopulations of cells. In addition, as I mentioned before, you need this copy number data as input, so you're really relying on how good that is. So really, kind of moving on from there as the field has moved forward, in the past few years, really the past, I think like 7-ish years, there's been really this huge explosion of these really powerful single cell technologies. So these single cell technologies are capable of performing some of the same analyses, but actually at the single cell level of resolution, which provides far more granular information than a lot of these bulk assays. And as these single cell assays have come online, particularly single cell RNA sequencing is one of interest. Along with that, certain bioinformatics methods have popped up in order to get a better sense of, to perform better subclone inference from this data. So there are a couple methods, one of interest from 2018, one from this past year, that actually they infer subclonal populations of cells from single cell RNA sequencing. So this is expression data at the single cell level. So you can picture, you have a sample that has thousands of cells in it, and for every cell you're getting, a quantified, amount of expression for, each of potentially thousands of genes, with some limitations that I'll get to. But these methods, they actually perform genotyping and pair it with expression. So if you picture a section of the genome of DNA, and that gene might get transcribed into mRNA, if there's some sort of a mutation or alteration in the DNA sequence, then this is going to be reflected in the mRNA sequence. So by using the single cell RNA-seq that is capturing those mRNA transcripts, you can actually find some of these mutations. So they pair this mRNA genotyping along with the expression data that you would typically use single cell RNA-seq for, perform some various statistical modeling of noise in the expression data, and then infer some of these subclonal populations of cells. One limitation to this, a limitation that it shares with the bioinformatics methods that operate on bulk data that I mentioned before, is that these are really limiting themselves to genetically distinct subpopulations of cells. While single-cell RNA-seq is an important assay of primary interest, there are really all these other single-cell assays that are starting to show up that are worth discussing. So, they can potentially be used to study this heterogeneity question too. DNA sequencing, so single-cell DNA-seq looks at the nucleotide sequence at the single-cell level. Single-cell RNA-seq, which I mentioned, looks at expression. You have things like single-cell combinatorial indexing ATAC-seq, which is it's looking for open regions of chromatin that are accessible for transcription at the single-cell level. You can look at methylation. So there's single-cell whole genome bisulfite sequencing, where you can actually look at the DNA methylation patterns in each cell. And then transcription factor target relationships with ChIP-seq, which I'm going to discuss transcription factors a little more, but these are essentially just genes that are transcribed into proteins, and then those proteins have behavior where then they bind to different places in the genome and affect the expression behavior of other genes. So with that idea of multiple single-cell omics assays that can be used to study the question of heterogeneity, I really just want to take a minute to discuss a really nice example of an analysis from 2 years ago that did use multiple single-cell technologies specifically to study this question of subclonal populations of cells capable of driving drug resistance. So this analysis, they used two different data types. So they used single-cell DNA sequencing, which they largely used for copy number analysis, looking for the amplifications and deletions of different genes. And they used single-cell RNA sequencing. And they applied this to 20 patients with triple-negative breast cancer, that difficult to treat subtype of breast cancer I mentioned before. And they were really looking for, in the patients that did not respond durably to therapy, was there some sort of subpopulation of cells that was present prior to therapy that was driving this resistance, or was it something that appeared later? So what they found is that there were 10 patients that had treatment response, but then In the 10 kind of clonal persistence patients, they found that they found two things. So they found that there was at least one subpopulation of cells that was genetically distinct that was selected for by the drug treatment, but then also the expression patterns shifted. So you can see on the left, you see these multiple subpopulations. Several of them respond, but then the bottom one, this is a distinct subclone that is actually selected for, and then this copy number genotype becomes present following treatment. But then interestingly, they found that these cells following treatment showed gene expression profiles that were not found in any of the cells prior to treatment. So this led them to conclude that not only was there selection for this subpopulation of cells capable of driving resistance, but then there was actually this transcriptional reprogramming process where these cells that were selected for were actually shifting their expression profiles in response to the drug. So this is just really just shows kind of the potential of, how these technologies might be able to be used to better understand this question. Now I'm just going to mention a few of the technological and the biological challenges that may arise when using these single cell data types to address the problem of tumor heterogeneity and drug resistance. So one of the key problems is the integration of multiple single cell data types. You can imagine a scenario where you use one data type and you potentially want to get even more information or bolster that analysis by adding in another data type. You might want to look at mutations and methylation, or methylation and expression, or something like that. The problem is, since these are destructive assays, even if you're taking a little bit of tissue for each analysis, all from the same sample, you're not performing on the same set of cells. So you don't have this kind of one-to-one mapping of, we have this cell and this is what the mutations look like, this is what the gene expression looks like, this is what the methylation looks like. And you kind of have to perform that mapping. So this is kind of an open and developing question in the field of how do you successfully integrate these different data types? Additionally, there are certain challenges that really are just inherent to the single cell assays themselves. The most prominent of which is this issue of sparsity and dropout. So, you really get this power of being able to look at things at the single cell level of resolution with, for example, single cell RNA sequencing, but you can't necessarily rely on having good signal at every site for every cell in the data set. For example, you might have a certain cell, and for a particular gene there might be no signal there, but that doesn't mean there are no mRNA transcripts inside the cell of that type, because you can have things like just inefficiency in capturing those transcripts, inefficiency in converting them to cDNA and amplifying them. And so, this dropout issue can pose a challenge. Additionally, you have uncertainty quantification, which Nate Evans did a really nice job of talking about in a lot of detail last week. And then also just data integration across multiple samples and experiments. So this is, you can think of controlling for batch effects when you combine, data from cells from multiple patients, multiple sites, multiple experiments, that sort of thing. There are also some biological challenges associated with this. So, you have this issue of transcriptional bursting, where, when you use one of these single cell assays, you're really just taking a snapshot in time. But if the gene expression of a particular site is not constant across time, there's some sort of fluctuation in a stochastic way, and you're just catching one time point, then you're not really fully capturing this whole dynamic change. You can have monoallelic expression, where a cell might only express one allele at a time. You can have spatial heterogeneity. This is one that, I mentioned the idea of multi-region sequencing before, and how you might have spatially distinct subpopulations of cells within a tumor. So if you sample one section of the tumor, you might be missing cells that are present elsewhere. So this shows a need for multi-region sequencing. And actually, one of those methods that infers subclonal populations from the bulk assays, one of the main conclusions of that paper was really a need for multi-region sequencing to fully capture the subclonal profile of a tumor. You also have cell state plasticity, the transient expression of different surface markers, which pose challenges. You have tumor purity, which is just the contamination of the cancer cells with the cancer sample with other cell types, so healthy cells and others. And then you also have one challenge that I'm going to talk about in a little more detail, which is really the need to study things from a systems biology perspective. And instead of looking at individual genes, looking at the gene regulatory networks and how their activity changes. So I'm going to talk a little bit more about this concept of gene regulatory networks. So rather than just looking at individual cells and how they're mutated and how they're expressed, really there's been a shift in the field to moving towards a more systems biology approach of actually looking for the different ways that interconnected networks of genes and proteins regulate each other. So we have this idea of a transcription factor and its target. So you have a transcription factor, which is just a protein that then binds somewhere in the genome and affects the expression activity of its downstream target. And these make up a network. So you can think of these transcription factors, and then they have these downstream targets that they affect. As you can see, a particular downstream target could be affected by multiple different transcription factors. And these networks are in healthy tissue. They're typically very carefully regulated. But if they become dysregulated, this can lead to aberrant gene expression patterns that can change cell behavior and actually drive tumorigenesis and cause a patient to develop cancer. Really, a main advantage of studying things from a gene regulatory network perspective is that this captures mechanisms that could be, it captures changes in these gene regulatory networks that could arise from multiple different mechanisms. So there could be genetic factors like a mutation that's causing the dysregulation of a gene regulatory network. That would be the more classical view, but this could also arise from other things like epigenetic changes and microenvironment-induced changes. And you're going to see all these changes at the level of expression in the gene regulatory networks. There's another, what's really just a side effect, a convenient side effect of using the gene regulatory network approach is that it addresses this sparsity issue. So, we want to study heterogeneity. The bulk assays are kind of lacking in resolution. So we move to the single cell assays, which gives us the increased resolution, which is really nice. But then it introduces this new problem of sparsity. But then by studying things from a gene regulatory network perspective, you can actually kind of take a more holistic approach and use, instead of just one gene or a small list of genes, you can look at this broader behavior, which might allow you to recover signal that you might actually miss if you look at it from a more kind of single gene centric perspective. In the past couple of years, there have been these methods that have arisen in the literature that have been developed to infer some of these kind of gene regulatory subnetworks. So you can picture a transcription factor and its downstream targets as forming a sort of a subnetwork or a regulon. So these can be, there are methods that will actually infer these from single cell RNA sequencing literature, for example, or data, for example. So there are different approaches that these can take. So one of these methods, scenic, it uses co-expression networks, and then it incorporates transcription factor binding domain information to narrow down the list. And then there's this other method, scribe, that takes a different approach where it uses kind of time-coupled gene expression vectors to look at how the expression of one gene earlier in time affects the expression of another gene later on. But regardless, there are these different methods that exist that have mostly been applied at a clonal rather than a subclonal level of analysis, but they do exist. I just want to take a minute to talk about this analysis that came out in just September of this year, where a group in China looked at some glioblastoma samples and actually attempted to perform gene regulatory network inference at a subclonal level of analysis. So really what they were doing here is just applying one of these previous tools, so scenic, that gene regulatory network inference tool I mentioned. They used that to look at these glioblastoma samples and to look for kind of this regulon, kind of gene regulatory subnetwork activity that was present at a subclonal level. So they did various things in this paper, but of primary interest, they took you can look at this figure on the right here. What they did here is that they took a glioblastoma patient sample from a single patient. So this shows a sample from a single patient. They actually broke it down into different cell subtypes using a classifier specific to glioblastoma. So then within one patient, within one subtype, they actually found multiple subclones. And really what you're seeing here is, on the right side, you have the list of these so-called regulons. So these, the name is really just the name of the transcription factor. And then on the right, you can see the number of downstream targets. So this is really just capturing the activity of this kind of regulon overall. And then you can see in the two blues and two reds is a little confusing, but basically the light blue and the light kind of pinkish red are two different subclonal populations of cells. And then the dark blue and dark red just show upregulation or downregulation of the expression of these regulons. So for example, you can see fourth from the top, this SOX10. You can see the first subclone shows downregulation of this regulon relative to the second one. And then, fourth from the bottom, FOXK1, you can see, this regulon is upregulated in the first subclone relative to the second subclone. So this, this is kind of a preliminary analysis, but it's really nice example of how you can potentially use some of these gene regulatory network tools to study what's going on at a subclonal level. But, you really then have to, there's some next steps that are needed to tie this to things like drug resistance. So with that, there's a lot more to be done in this field. There's a lot more that needs to be done in order to solve these problems, including the detection of resistant subclonal populations of cells that may or may not be genetically distinct. They may arise from other factors. If these can be identified prior to therapy, this can lead to, yes, potentially the development eventually of new therapies, but also just different clinical decisions based on a better understanding of who will and will not respond durably to certain treatments. Another challenge is the tracking of clonal evolution, so looking at how these different subpopulations of cells change longitudinally throughout the course of treatment. There's a need to better understand the types of heterogeneity that are present within a tumor, their mechanistic causes, their clinical consequences, and then really lastly, the inference of subclonal gene regulatory network activity that is specifically associated with drug resistance. So just going back briefly to that, this last analysis, one of their major findings here was actually that they saw more inter-tumor heterogeneity, so differences between patients in this regulon activity than they did intratumor heterogeneity, meaning that there's a lot of genetic background that is just specific to that particular patient. So, detecting subclonal gene regulatory network activity overall is nice, but it's really important to zoom in on the subset of that activity that particularly is associated with driving drug resistance and treatment failure. So this could be done by novel applications of existing tools or potentially development of new tools in order to track and better understand these resistant subpopulations of cells that are responsible for driving drug resistance, treatment failure, and really just the fact that the state of cancer treatment today is, we don't always have a durable therapy that's effective for every patient. With that, I would like to acknowledge my advisor, Dr. Guangming Wu, some other professors who have helped me, Shannon McQueenie, Aaron Cohen, the DMICE students, everyone who's helped me, listen to my practice talk and get this together, the DMICE faculty and staff who are invaluable as always, some other people who've been really helpful in helping me get ready for this, and then the funding through the National Library of Medicine. And with that, I would like to conclude and open it up for questions.
Great, thanks. There's a question in the chat from Ted.
Okay. I can't see the chat. Let me open it up.
Yeah, okay. Or you could stop your screen sharing.
Mm-hmm. Just right now, before I forget. Okay. Have you considered using cell marker expression from flow cytometry methods? Yeah, so that's an interesting thought. I've definitely have thought about that. One challenge with, I mean, these flow cytometry methods can be really useful. They also come with a couple challenges. One of them is that the really just the number of markers that you can look at once is much lower. So, you might be looking at, I don't know, 20 or 40 or 60 genes rather than thousands. So that's kind of a limitation. But, these can be useful certainly for identifying, different cell subtypes and that sort of thing. It is, you mentioned kind of the transient expression of different surface receptors and kind of just dynamic. So this really, oops, sorry, this one have a, oh, okay. So this really comes up, with the cancer stem cell question. For example, you're relying on kind of consistent and continuously expressed surface markers, which can have its limitations. But that's definitely something to consider. And I think some of these flow cytometry methods can be really useful. So yeah, thanks for the question. Useful.
I have a question. Can I ask, or do you want to just do it from chat, Bill?
Oh, no, go ahead.
Go ahead and ask.
So nice job, Chris. One thing that comes up when we're thinking about, methods and applications is the timing right? Do you feel like from your review of the literature that there's sufficient data for this kind of direction that you want to go in? I mean, you kind of hinted the areas of interest and think about that kind of last, gap slide, right? So do you feel like all of those are feasible in the near term?
Yeah, I mean, I've taken a little bit of a look at data, and I'm definitely going to be getting a lot more into that in the next few months to see what's feasible. I think that there's definitely more that can be done with what's out there right now. But I think a key part of this is going to be, for example, if I use patient data, looking for data that actually is tied to data about meaningful clinical outcomes, and then to kind of be able to tie it to the question of biological interest. So that's definitely something that I'm continuing to think about. But
yeah, I think my only comment would be depending on the final approach, and this is just your symposium, so it's not a critique, it's just more of a thought question for you to think about as you're writing your proposal. Some of the methods are going to be very data intensive, and especially for network inference, potentially depending on the direction you go. And so I think that is kind of a key. I don't want that to drive the question, but as you're kind of doing your feasibility assessment for your proposal, just keep that in mind. It was very nice that you had a number of topic areas, but not all of them will be ready in the near term. Does that make sense?
Right. It does, yeah. And that's a really useful comment. And one thing that I was kind of looking at is some of these methods try to kind of use workarounds to study some of these questions. So there are some of the methods that actually they infer the genetic subclones from the single cell RNA sequencing data, but then if you have other methods that are using the expression data to identify the subclones, and then you look for things like different gene expression. You look for differential expression between subclones, but then you defined those subclones using the expression data. It's kind of a self-fulfilling prophecy. So it's I think the methods are kind of at this point that makes sense that they're kind of constrained by what data are available and the technologies that are available. So it's definitely something I need to keep thinking about. I have a question. Oh, sorry, go ahead.
I was just saying you have 2 new questions in the chat, FY.
Yeah, okay. Okay, so it looks like one from, here we go. What methods are you considering for identifying potentially resistant subpopulations? To follow up from Shannon, which might have data available. Right, so one thing I've thought about doing is looking at more like cell line type data that has expression data along with and it also has the availability of like drug response data, and then kind of pairing that with any sort of patient data that I can get. But again, at this point, looking at what data are available is going to be my next step. And I didn't want the availability, what data were and weren't available to drive the question, because I wanted to make sure it was of clinical importance. But that's from a feasibility standpoint, that's definitely something that I need to be thinking about. Does that answer your question? I can.
There was also a question from Nate Evans.
From Nate, yeah. Okay. Yeah. So from Nate, how are you planning, or how have people done it in the past, to link sub to drug resistance or sensitivity? Right. So this is, if you want a more complete answer to that question, then feel free to show up in a few months when I'm doing my dissertation proposal. But at this point, I have a few different ideas of how this could be done. One thing that I think it's worth mentioning is that subclones are usually defined in different ways depending on the question of interest. So some analyses will identify subclones as being genetically identical. Some will look at just a list of a few genes of interest, like oncogenes or something. So I think one thing that that I really want to do is to figure out how to define subclones based on, kind of in the context of the question of interest. So to kind of be able to divide up subpopulations of cells within a tumor based on their activity, not of all, for example, gene regulatory networks, but specifically of the gene regulatory networks that are relevant to the drug response, which, the key question is, okay, where does that come from? And like I mentioned, depending on, if there are limitations of what data I can get that's tied with clinical outcomes, I might be able to use cell line data that does have more clear response information. I think, okay. Yeah, I think I answered the questions in the chat. If anyone else says.
Give people another few seconds to ask more questions, either verbally or in the chat box. I can just, as someone who works with knowledge access systems, once you perfect some of these techniques, I see all sorts of interesting follow-on work with information retrieval systems when you identify a patient that has some sort of clone that's growing and we don't know the therapy to, et cetera. Should be interesting. I too will look forward to your proposal defense down the road.
Yeah, well, thanks. And I kind of, think about this as, the people in our department were kind of arrayed along the spectrum from, more basic to then translational research to really clinical. And, this probably fits somewhere in the more translational realm, but then, there's an idea of, just trying to understand the biology that can then be used to then kind of implement clinically later on.
Yeah, good. Okay, well, thank you, everyone, and have a good day. See you all later.